Exploring Life. Chapter 21, Human Evolution. Part 3. Humans reproduce sexually, following a reproductive cycle that is deeply rooted in our eukaryotic ancestry. The human reproductive system shares many features with other placental mammals, reflecting our common evolutionary origins. It consists of a coordinated network of organs that support gamete production, fertilization, and gestation. In placental mammals like us, the developing embryo is nourished and sustained through the placenta. Together, these systems ensure the continuation of the species, from the formation of haploid egg and sperm to the development of a new human life. Sex is determined at conception. Sexual development begins early, during the embryonic stage, when reproductive organs first start to form. By birth, females already carry all the immature eggs they will ever have. In contrast, males don't begin producing sperm until puberty. Puberty triggers a surge in hormonal activity, leading to the development of secondary sexual characteristics, like deeper voices in males or breast development in females. These changes shape the distinct physical features of adult men and women and signal reproductive maturity. In humans, menstruation is the monthly shedding of the uterine lining when fertilization doesn't occur. It's relatively rare among mammals, found mainly in some primates and a few bat species. This may be linked to our complex genetics. Up to 75% of human pregnancies do not develop properly and end in spontaneous abortion. Menstruation helps ensure that only healthy embryos are supported, giving them the best chance to survive. Like other animals, humans have specific windows in the ovulatory cycle when fertilization is most likely. But unlike most mammals, we don't advertise it. Humans exhibit concealed estrus, meaning there are no obvious signs to indicate when a female is fertile. This hidden timing may have influenced human behavior by encouraging long-term pair bonding instead of brief, timed mating. Human infants are born in a relatively immature and dependent state. They typically can't walk until around 12 months and remain unable to feed or care for themselves for years. Even among primates, humans show major delays in reaching milestones. Puberty begins at 10 to 14 years in humans versus 6 to 9 in chimpanzees. First reproduction is often delayed until the late teens or 20s, compared to 10 to 13 in great apes. Brain development continues into the early 20s, with the prefrontal cortex, responsible for decision-making and impulse control, maturing last. One theory attributes all of this to neoteny, a process that not only delays development but also retains juvenile traits like body proportions, playfulness, and low aggression, which may enhance sociability. This extended growth period requires long-term parental care, but also allows for the formation of deep, lasting social bonds that define human societies. Average global life expectancy is around 72 to 73 years, shaped by healthcare and nutrition. Menopause typically occurs near age 50, meaning many women live decades beyond their reproductive years. This extended post-reproductive lifespan is a unique human trait. In most animals, lifespan aligns with fertility. Females often die soon after they stop reproducing, conserving resources for the young. The grandmother hypothesis offers one explanation. Post-reproductive women improved the survival of descendants by providing care and support. By helping their kin, grandmothers gained evolutionary advantages that outweighed any competition for resources. Today, much of this caregiving is outsourced, but the evolutionary pressure for longevity remains. As childbirth is increasingly delayed into the late 30s or beyond, lifespan-linked reproductive health may shape who reproduces successfully, potentially driving human longevity even further. The human nervous system, the body's primary communication and control network, follows the basic vertebrate blueprint. It consists of the brain, spinal cord, and peripheral nerves. The central nervous system, CNS, includes the brain and spinal cord. The human brain, notably large for our body size, interprets sensory input, coordinates responses, and supports complex processes like thought, memory, and emotion. The peripheral nervous system, PNS, consists of nerves branching from the CNS to the rest of the body. It relays signals between the brain, spinal cord, muscles, organs, and sensory receptors, ensuring fast, coordinated function throughout the body. Even though the human brain is larger than that of most other vertebrates, its overall structure remains consistent. It's divided into key regions, each with specialized functions. At the top is the cerebrum, the largest part, responsible for decision-making, voluntary movement, language, and interpreting sensory information. Beneath it lies the cerebellum, which coordinates movement and helps maintain balance and posture. At the base are the diencephalon and brainstem, which control vital automatic functions like breathing, heart rate, digestion, and more, keeping the body running without conscious effort. What sets the human brain apart from other mammals, especially primates, is the size and complexity of its regions. The image on the left highlights the four lobes of the cerebrum: frontal, parietal, temporal, and occipital, each responsible for specific functions essential to human behavior and cognition. The image on the right reveals deeper brain structures. The hippocampus is brown, is key to memory formation and emotional regulation. Nearby, the basal ganglia, blue, help control movement, form habits, and coordinate motor activity. The human nervous system stands out in the animal kingdom not only for its size and complexity, but also for its unique traits and specialized regions. One striking feature is extensive cortical folding, grooves, sulci, and ridges, gyri, that expand surface area and support advanced functions like language, memory, and problem-solving. While other mammals also have folded cortices, human folding is unmatched relative to body size. Another key distinction is our reliance on vision. In some animals, nearly all cortical processing is devoted to smell. In contrast, over 40% of the human cortex handles visual input, reflecting a shift away from ancestral dependence on olfaction. As upright posture raised our heads farther from the ground, smell became less essential. By mammalian standards, our sense of smell is poor, but it hardly mattered. We weren't sniffing tracks, we were hunting in daylight, scanning the horizon, and running prey into exhaustion. Some brain regions aren't just unique, they help define what it means to be human. At the front, the prefrontal cortex governs decision-making, planning, and impulse control. It helps us think ahead, manage emotions, and navigate social life. Damage here can dramatically alter personality. Broca's area controls speech production, while Wernicke's area handles language comprehension. These regions work alongside the muscles used for speech and the ears to make complex communication possible. But these abilities depend on early experience. Without exposure to language and social interaction, especially before age 7 or 8, the brain may never fully develop them. A child raised like Mowgli might survive, but without early human contact, they'd lose language and connection. If that loss became global, even for just 2 generations, our knowledge, values, and institutions wouldn't just fade. Civilization would collapse. The nervous system controls 2 main types of behavior: reflexes and learned responses. Reflexes are fast, automatic reactions, like the suckling reflex in babies, which helps them feed without thinking. These built-in responses protect us from danger, like pulling your hand away from something hot. Learned responses are different. They develop through experience. We remember what worked or didn't work and adjust our behavior. This ability to learn and change is a big part of what makes humans so intelligent. Some learned behaviors, like eating when we're hungry or drinking when we're thirsty, feel good when we do them. That good feeling teaches us to do them again. One of the most common explanations for addiction is that drugs trick the brain by copying that same good feeling, even when there's no real need. That's what makes addiction so powerful. It hijacks the same brain mechanisms that have helped us survive for millions of years. Some parts of the human body are evolutionary leftovers, features that once had important functions but have since been reduced or repurposed. These are called vestigial traits. Examples include the coccyx, or tailbone, a remnant of the tail our distant ancestors used for balance and movement. The appendix, once involved in digesting tough plant material, now plays only a minor immune role, if any, and is often removed when inflamed. Some parts of the human body are evolutionary leftovers, features that once had important functions but have since been reduced or repurposed. These are called vestigial traits. Examples include the coccyx, or tailbone, a remnant of the tail our distant ancestors used for balance and movement. The appendix, once involved in digesting tough plant material, now plays only a minor immune role, if any, and is often removed when inflamed. We also retain vestigial reflexes, automatic responses that once helped our ancestors survive. One example is the palmar grasp reflex in newborns. Place something in their palm, and they'll instinctively grip it. For our primate ancestors, this reflex helped infants cling to their mother's fur while she moved. Another is goosebumps, tiny muscle contractions at the base of each hair follicle, triggered by cold, fear, or surprise. In our fur-covered ancestors, this reaction fluffed up their coat for warmth or made them appear larger to threats. Today, these reflexes offer no real benefit, just a fascinating glimpse into the evolutionary past written into our bodies. Atavisms are rare, fascinating traits that reappear after being absent for many generations, tracing back to ancient ancestors. One striking example is the presence of a tail-like structure in newborns. Though extremely rare, this reflects our evolutionary past when tails were functional. Another atavism is the occurrence of extra nipples, often found along the milk line, a remnant of our ancestral pattern of having multiple mammary glands, as seen in many mammals. This image shows an example of atavism, the presence of wisdom teeth, also called third molars. Once useful for grinding tough plant material, these teeth are now often problematic. As human jaws became smaller over time, many people no longer have room for them. Some have all 4 wisdom teeth, some have fewer, and some never develop them at all. In this X-ray, you can see misaligned wisdom teeth on the right, pushed out of place by reduced jaw space. It's a striking reminder, evolution doesn't delete old traits, it buries them. And sometimes, they resurface. Head hair reflects both biological and social influences. From staying cool in the sun to signaling personality, health, or youth, our hairstyle choices may be shaped by more than we realize. What drives different human haircut choices? Let's hear from you. Certain genetic mutations can bring ancient traits back into view. Hypertrichosis, for instance, causes excessive hair growth even in areas usually hairless in modern humans, possibly reactivating dormant ancestral genes. Another example is microcephaly, a condition where the head is unusually small. In some cases, its proportions resemble early hominins. Though rare and medically significant, these traits offer a glimpse into the deep evolutionary blueprint still etched in our DNA. Our bodies came from this planet, built from its elements, shaped by its forces, and carrying its history in every cell. We are driven by biology, instincts, reflexes, ancient adaptations. But we're not defined by biology alone. Language, memory, love, curiosity, these aren't coded in our genes. They're passed from mind to mind, generation to generation. And that's what makes us human, not just what we are, but what we choose to become. Check your understanding, a chance to pause and reflect on what you've learned. Take a moment to organize your thoughts, or jot down a few notes before moving forward.